So there are two major groups of genes which cause cancer. Those are the tumor-suppressor genes and the proto-oncogenes. And then there are some smaller groups of genes which are involved or mutated in part of the multi-step process that also contributes to cancer. And those are genes which encode DNA repair proteins and enzymes, genes that regulate telomerase, genes that promote angiogenesis or new blood vessel growth, and then the genes needed for cytoskeleton or extracellular matrix structure. And those genes are really important. When they mutate, that's when a cell is able to leave its primary location and move to other locations or become malignant. But the two major groups of genes that we're going to focus on are the tumor-suppressor genes and the proto-oncogenes. The tumor-suppressor genes encode the proteins which, when they're functioning properly, will stop the cell cycle at appropriate times. The genes which encode proteins which promote the cell cycle appropriately are called the proto-oncogenes. And so mutation of tumor-suppressor genes or proto-oncogenes is where you start to have problems, where you start to lose control of the cell cycle. Now the tumor suppressor genes, there's many of these, but for each one of these tumor suppressor genes you have two copies, one on each of the two homologs. And normal people are homozygous for a normal copy of those genes. And the tumor suppressor genes, when they're functioning correctly, you get normal cell divisions. But if you get a mutation and both copies of the same tumor suppressor gene in the same cell, then you end up with a homozygous mutant case, and that cell will start proliferating in an uncontrolled way. So the tumor suppressor genes are sort of like the brakes on a car, but you have two copies. So if one gets mutated, you still have the other copy, you're still okay. But if both copies get mutated, if neither copy is working, then you'll lose control of that cell cycle. Now you can either get a mutation in both alleles, or you can get a mutation in one allele, shown here with a little a, and then a deletion of the other allele. That would cause the same effect. And when you have a deletion that removes one allele and then a mutant allele on the other copy, this is usually called loss of heterozygosity, or LOH. That indicates that there was a deletion. You had a heterozygous individual, and then a deletion of the good copy of that gene led to a cell that has no good copy of that gene, and that cell would become the original cell that starts the tumor. Now, some tumor suppressor genes that you should be aware of, that you are aware of course, RB, the retinoblastoma gene. Remember, the two-hit hypothesis said that you have to have both copies of the RB gene have to be mutant in the same cell, and that would start the retinoblastoma in the eye. Another gene that we'll talk about is called P53. This is also a tumor suppressor gene, and it's important, pretty much involved in more than half of all cancer types involve a mutation in P53 as part of the multi-step progression to cancer. Then some other ones, the neurofibromatosis gene and the Wilms tumor gene here. But there's actually many of these, but these are two that we're going to use as our main examples. The P53 that we'll talk about in a minute, and then the retinoblastoma gene, which we've already discussed. So P53, it's kind of a weird name for a gene. It's actually a typical name for a protein. They typically name proteins when they're too lazy to make up an actual name. They often name them after the molecular weight of the protein. So P53 means a protein that weighs, or that has a mass of 53 kilodaltons. But it's not just any protein that weighs 53 kilodaltons. It's this one specific protein that weighs 53 kilodaltons. And this molecule is a tumor suppressor. And about half of all human cancers have a mutation in p53. So it's a really important tumor suppressor that is often mutated. And in 1993, Science, which is one of the premier journals, chose p53 as Molecule of the Year, which is kind of funny because I bet you didn't know there is a Molecule of the Year. But anyway. Now, one thing that's interesting about p53 is not only is it involved in a lot of different cancer types, mutation of p53, but there's actually a syndrome. It's called Li-Fraumeni syndrome. It's where you inherit a dominant p53 allele, and you end up with an individual who has a dominant p53 mutation in every cell, and basically they can get tumors from any of their tissues. So you get these massive, cancers in all types of different tissues all over the body. So it's very rare, but it's also very horrible. So what does a tumor suppressor do when it is acting appropriately? Well, I'll give you the example of the retinoblastoma protein, which is copied from the retinoblastoma gene. The retinoblastoma protein, when it's not mutated, it binds to a protein called E2F. All right? And E2F is a transcription factor that's needed for transcription of genes for S phase. So when the RB protein binds to E2F, it holds the cell back from going into S phase. Now during the cell cycle, there's a buildup in between, or during the different phases, there are buildups of proteins you may have heard of called cyclins and CDKs. And the increase in concentration of cyclin-D and cyclin-E with their corresponding CDKs results in them being activated, and they will phosphorylate, which means put phosphate groups onto the retinoblastoma protein, which changes its conformation and causes it to release its hold on E2F. And then E2F is released, and it activates transcription of genes needed for S phase. So this is all the proper functioning of retinoblastoma. When the cell is not ready to go into S phase, the retinoblastoma holds on to E2F. When the cell is ready to move on into S phase, the retinoblastoma protein releases E2F and things proceed. So what would happen if the retinoblastoma protein is not produced or is not functional at all? Then if you would take the retinoblastoma protein out of this picture, the E2F would not have anything to bind to it. And when the E2F is not bound to anything, it is active. And so E2F would inappropriately push the cell into S phase, even when the cell might not be ready, or the cell might need to stop and do some DNA repair. But if retinoblastoma protein wasn't there, the E2F would be able to push the cell forward when it's not the right time. And so that is one of the examples of kind of a loss of control of the cell cycle. Now, oncogenes typically act in a dominant way. Tumor suppressor genes typically act in a recessive way, although p53 can be dominant or recessive. But most other tumor suppressor genes are recessive in that you have to have the two hits. You have to lose both copies of a tumor suppressor gene in the same cell to start uncontrolled proliferation. But oncogenes typically act in a dominant way, meaning when you have, there are many oncogenes, and when you have the normal copies of the oncogenes are called the proto-oncogenes. So that's the non-mutated genes. Proto-oncogenes, you have two copies for each of them, and they normally produce growth-stimulating factors. They normally push the cell forward in the cell cycle when that's appropriate. If you get a single mutation in either allele, and it has to be a very specific mutation, you can result in one allele that becomes hyperactive. Or we should say the protein that it encodes is hyperactive. A hyperactive stimulatory factor will basically be overactive. That's what hyperactive means. So an overactive stimulation of cell division pushes the cell forward in cell division even when it's not supposed to. So that's why one specific mutation in proto-oncogene creates what we call the oncogene, which is the mutant form, and that pushes the cell then, the protein product of that gene pushes the cell forward in the cell cycle. So the outcome is really the same as with the tumor suppressor gene. You're pushing the cell forward, or the cell is moving forward in an inappropriate way too quickly through the cell cycle. And there are a lot of oncogenes. RAS is an oncogene, MYC is an oncogene, and typically when it's a cellular copy of an oncogene, you'll see a C in front of it. So, for example, like if I said C-RAS, that means it's the allele in the cell. C-MYC would be a cellular copy of that gene. So when you have a C in front of it, that means cellular. Okay, and that can be mutated. That can become mutated and act in an inappropriate way to push the cell through the cell cycle. About a third of all tumors have a RAS mutation, so that's pretty common. All right. Now you can also get oncogenes that already have a mutation that come from a virus, and those are called viral. So a viral RAS would be a copy of the RAS gene carried by a virus that infects your cell, and that RAS gene has already been mutated. So when it comes into the cell, it acts in a dominant way and takes over and starts pushing your cell through the cell cycle too quickly. But that would be viral RAS. All right. Now, there are certain point mutations that are associated with these genes, both tumor suppressor genes and oncogenes. If they get certain point mutations, they can lead to cancer. But there's also certain chromosomal rearrangements that are associated with cancer. And one of those is shown here. This is called, the product of this translocation is called the Philadelphia chromosome. And it's found in unrelated patients who have the same kind of cancer called chronic myelogenous leukemia. So you probably know leukemia is a white blood cell, cancer of the white blood cells. So it's a specific type of leukemia. And what they find is that in the cancerous cells, a lot of the patients, a certain number of the patients have the same, strangely, the same translocation. So if you have, for example, here's a gene, CABL is the cellular, it's a proto-oncogene, a normal copy of a proto-oncogene. And over here you have a gene called BCR. Okay. And what happens is the proto-oncogene by translocation is moved to the other chromosome. So it's a chromosome 9 to chromosome 22 translocation. And what happens as a result, these two genes end up sort of being spliced together in a weird way that you really can't see from this picture. But the mRNA that's copied from this ends up including parts of the BCR gene and parts of the ABL gene, and you end up with what we call a fusion protein, a single protein that fuses what should have been two genes together. And that fusion protein functions in an overactive way, and so really what you have is an overactive oncogene. And so an overactive oncogene will give you cancer. So one cell has this translocation happen inside of it, and that cell is the one that starts losing control of the cell cycle as a result. All right, now this is a different one. This is found in Burkitt's lymphoma, and it's another type of lymphoma, and leukemia are similar. They're white blood cell, cancers of white blood cells. And this translocation moves a different oncogene. The MYC gene is moved to an area. If you look here, the MYC gene is really in one of these dark areas, and so it's not expressed overly much. Remember, the dark staining areas have a certain amount of transcriptional repression. So when that gene, which is right in that area, is moved into this light-colored area, you end up with an overexpression of the c-MYC gene. So the gene itself doesn't mutate, it just is expressed too much. So overexpression of a perfectly good gene gives you too much stimulation of the cell cycle. So that's a different one, but it's the same concept, any kind of overexpression or overactivity of the protein. So those are things that can happen in your cells, translocations or point mutations. Then, I touched on this, viruses can also bring in cancer genes, which adds a whole nother layer to this. So you may be familiar with some of these viruses. Human papillomavirus, that's been getting a lot of press. The HPV vaccine that's available fights or inoculates against this virus. Hepatitis B virus, which causes hepatitis. T-cell leukemia, virus 1 and 2, Epstein-Barr, and herpes virus. So these viruses can work in different ways to cause cancer. Sometimes they carry mutated oncogenes. And when they deliver that oncogene into the cell then expresses that gene. But they can also just cause problems because they integrate onto chromosomes. And so you see the different types of cancer that can be caused by these. So the way that they can cause cancer is either, I'm going to show you, it's really right down here. Either the virus carries, this little spot here is a mutation on the oncogene. The virus brings in a mutated oncogene which integrates onto the host chromosome, and then the cell expresses that mutated oncogene and it becomes cancerous. The other thing that can happen is the retrovirus might not carry an oncogene, but the viral chromosome, when it integrates onto your chromosome, viruses have very strong promoters. That means they attract RNA polymerase strongly so that they can get a lot of expression of their genes quickly in the host cell. Remember, the host cell might be trying to fight this off, the virus is trying to win. So the virus have very strong promoters. Well, if that strong promoter inserts on a chromosome near to one of your normal proto-oncogenes, it can lead to overexpression of that proto-oncogene, and overexpression, too much of a proto-oncogene, leads to cancer.